Okay, great. So, welcome everybody. Okay, let's get going. So I want to say thank you all for joining us for our live Lipid Maps webinar. Today it's going to be presented by our guest speaker, Denise Biagini from the University of Pisa in Italy. It's entitled Detection and Quantitation of Oxylipins and PUFAs in Non-Conventional Biological Fluids, Potentials and Pitfalls. My name is Valerie O'Donnell. I'm based at Cardiff University in the UK, along with Thierry Durand, who's going to be introducing Denise, and he's based at the IBMM in Montpellier in France. Today's webinar is being sponsored by Cayman Chemical. We're grateful for their ongoing support, and now I'm going to hand over to Thierry Durand, who's going to introduce our guest speaker today.
Thank you, Valerie, and it's my pleasure to introduce our speakers. So Denise Biagini. Denise obtained her master's degrees in analytical chemistry, cum laude, in 2016 at University of Pisa in Italy. From November to 2016, she was a PhD student in chemistry and material science at the Department of Chemistry and Industrial Chemistry. She obtained her PhD, again, cum laude, and in May 2020, she is now a researcher at the Department of Chemistry and Industrial Chemistry of Pisa. Her research is mainly focused on the development of MS-based analytical method for health and disease assessment in conventional and non-conventional body fluid, plasma, saliva, breast, sweat, and DBS. In the recent years, she has been working on targeted metabolomic by tandem mass spectrometry for the determination of lipid mediators as marker of oxidative stress and inflammation in different research fields, including the COVID-19 pandemic crisis. And during her PhD, she spent 3 months at the research hotel of the Metabolomic Innovation Center in Edmonton, Canada, under the supervision of Dr. Rupsari and Mandel and Dr. David Wishart, deepening her knowledge on untargeted metabolomics. She is a co-author of 52 articles and an international peer-reviewed journal, and then more than 20 contributions of national and international congresses.
Hi, everybody. First of all, I'd like to thank Valerie and Thierry for their warm introduction and Lipid Maps for this great opportunity to share my research here today. So today I'll be focusing on the detection and quantitation of oxylipins and PUFAs in unconventional biofluids. An area that's quite exotic compared to the more traditional serum and plasma analysis. So I'll skip this brief introduction since Thierry did it perfectly. So as he mentioned, our team specializes in developing and validating LC and GC-MS-based methods for health and disease assessment, and we mainly explore unconventional biological fluids. What about me? Since 2016, I've been dedicated to oxylipins for different kinds of clinical applications. So my journey began with this research project based on the detection and quantitation of 8-isoprostaglandin F2 alpha, 8-isoprostaglandin E2, and prostaglandin E2 in red blood spots from premature babies. And what started with 3 analytes has now expanded to a panel of around 60 compounds among oxylipins, so oxygenated metabolites of polyunsaturated fatty acids, and their precursors. We can say that I fell in love with oxylipins, so I started with 3, and then I moved to a broad panel of compounds. We can say that, put in another way, I got stuck in this very exciting and fascinating research, but even very, very tricky research area. And so we expanded our methodology and thus covering a broad range of biological fluids and a broad range of clinical applications, including COVID-19, including heart failure disease and sports activities. So, what about oxylipin detection and quantitation? As you probably know, the 80 to 90% of the procedures for oxylipin analysis focus exclusively to plasma and serum. When it comes to unconventional biological fluids, things change a bit. Generally, the most used is urine that appears in 5 to 10% of the manuscripts. Cerebrospinal fluid follows with 1 to 3% of the papers reporting its use, in particular in neurodegenerative and neurological diseases. And then saliva, breast milk, and other biofluids that appears in less than 5% of the papers. So to sum up, as you can see here, the analysis of oxylipins in these unconventional biological fluids is still limited. So why choosing unconventional biological fluids? For sure, because collection is quick, non-invasive, painless, and that's great for the patient. There is no need for trained personnel, and this is ideal for self-monitoring, at-home testing, or repeated sampling. There's a reduced risk of infection and contamination, and that's great for biosafety reasons. And there's less need for cold chain storage, though it is not always true, as unfortunately in the case of oxylipins. So this aspect has to be checked carefully, analyte per analyte. And last but not least, we have to say that these unconventional biofluids show a great promise for point-of-care diagnostics without expensive lab infrastructure, and that's, we can say, the most important advantage. But unfortunately, there are lots of and many drawbacks in using these kind of biofluids. For sure, the lower biomarker concentration in these alternative biofluids, because compounds may not transfer or diffuse into these alternative fluids, or may partially diffuse into these alternative fluids. As it happens in saliva, for example, only the free fraction can pass through the salivary gland membrane and then move from the bloodstream to saliva. And it depends on the physical, chemical properties of the compounds and on his characteristics. So generally, you need eye-sensitive techniques. And there's a very high variability and inconsistency, even higher than what we generally observed in plasma and serum, and the presence of lots of confounding factors, as for example, hydration in the case of saliva sampling, humidity in the case of dried blood spot collection, or emotional state in the case of all these biological fluids. An high risk of contamination as regards saliva, for example, coming from food, drink, smoking, in sweat coming from personal care products or dirt, and breath. For example, the contamination coming from pollutants in the ambient air and diet. There's the lack of standard protocols, and it is truth even in the case of plasma and serum, but in this case is even worse. So we are so far from harmonization and standardization of analytical protocols in this area. And last, we should assess correlation of the concentration that we find in these biofluids with the concentration that we measure in the gold standard, that is plasma. This is required if you want to choose and use these unconventional biofluids as valid alternatives to plasma. So one of the most famous, together with urine, among these unconventional biofluids is for sure red blood spot, since its first use for newborn screening in the 1960s. It consists in the deposition of a drop of blood onto a filtered paper, and this drop of blood is then left dry for 2-3 hours and then extracted. Generally, a Batman-free protein saver card is used and a non-volumetric sampling approach. What does it mean? That generally you don't know the volume of blood that you are spotting on the paper, and then you perform a punch of a fixed diameter on the center of your spot, and you use this punch with a fixed diameter to perform your LC-MSMS analysis or GC-MSMS analysis. But what's the main issue and the main concern in using a non-volumetric sampling approach? That sometimes you may have some sampling effects that can affect your concentration in DBS, and they mainly related to hematocrit effect, which is related to hematocrit levels, so the percentage of red blood cells that you have in your blood. And this means that if you have a different hematocrit, your blood is more or less viscous, and thus it diffuses more or less easily through your support or your card, and you will have an inconsistent volume of blood into the disc of a fixed diameter that you punch from the center of your spot. And another effect is the chromatographic effect. So it is related to an inhomogeneous distribution of the analyte across the spot. So generally you have a lower level of concentration of the analyte in the center and a higher concentration in the peripheral area. So generally, when you are dealing with this kind of sampling effects, in order to ensure an accurate quantitation of your analyte, you have to move to a volumetric sampling approach. This means that you know exactly the volume of the blood, and you then extract the entire spot for the LC-MSMS based analysis. For these purposes, you may use some of these volumetric devices. For example, this microcapillary device, that is a volumetric microcapillary device from Sarsted, the Minivet POC, that handles from 20 to 100 microliters of blood. And then once you collect the blood, you directly spot onto your card. There's the EMA-Spot device, in which the absorbent paper is housed inside the device together with the secant in order to speed up the drying time. And last, there are these microfluidic systems that are the best choice if you want to be accurate and precise in your quantitation, as for example Capitainer B or Emaxis DB. The main concern, especially in the past, was about the sampling volume. So they were only available with very low sampling volume, as for example 10 to 20 microliters, that honestly should not be enough for this kind of approach for as concern oxylipin detection and quantitation. And what about saliva and or oral fluid? Because we generally speak about saliva, but it is not so correct, because saliva refers to the fluid that generally comes exclusively from the glands that are present in our mouth, so as the parotid gland, the submandibular gland, and the sublingual gland. Instead, oral fluid consists of the fluid coming from the glands plus the contribution of microorganisms and of oral mucosa. And that's more correct, since in general we collect the whole fluid that comes from our mouth. So generally to sample saliva or oral fluid, I will talk about saliva because it's generally more common and it's more, we can say, generally speaking, it's more common referring to saliva. So as regards sampling, you can use passive drawing. In this case, you can collect an uncontaminated and unstimulated saliva by drawing, so by collecting your saliva in your mouth and then drawing saliva through this cup. Then saliva is collected into this tube and is ready for the extraction and analysis. This kind of approach, it's a bit annoying, unfortunately, and the main issue is even related to the viscosity of the fluid that you can obtain, and that makes very difficult the extraction. So another kind of sampling device and sampling approach may be the use of swab. So this is generally employed for RNA and DNA detection. The main concern here is about volume. So the volume is a bit too low and so may not be desirable in case of ultra trace or trace analysis, as in the case of oxylipin detection and quantitation. Another kind of sampling device may be the salivate device, which is generally employed for cortisol collection and analysis. Generally, salivate is made up of a swab that is bigger than this one, so it can absorb from 1 to 2 ml of saliva. Then, once you collect saliva, and you can collect both at low stimulation by putting in your mouth and leaving it in a fixed position, or you can roll in your mouth, and in this way you can collect stimulated saliva. And then once you have collected the sample, you can put the swab into the device, perform a fast centrifugation, and then collect your fluid. So which kind of device or which kind of sampling approach is it better to use? It depends. It depends on your analyte. It depends on the characteristics of your analyte and on your application, on the types of patients you are dealing with. So it depends if you have to collect samples from babies or from adults or from newborns. So in general, you have to take into account that even in the case of oral fluid, there are sampling effects that can affect your concentration in these biofluid, and in particular that can change the concentration and makes it not and hinder the correlation between the concentration of the compounds in saliva compared to blood and to plasma. Generally, it depends, as I said, from the molecular weight of the compounds. It depends on its pKa. It depends on his lipophilicity, and in the way the compound transfer from the bloodstream to saliva. So there are different ways of transfer, as for example, passive diffusion, active transport, or paracellular route through tight junctions. So it depends on the characteristics, and this makes the compounds going easily or slower or faster from the left side to the right side, and this makes the compound more or less affected by changing in the sampling flow rate, for example, or pH. So as you can see here, indeed, a change in the flow rate, for example, may induce a change in the concentration of bicarbonate ions and thus a change in the pH. So you have to be aware of that, and you have to know which is the pKa of your compounds, and you have to know if your compound is in a neutral state or charged state, and if this increase in the flow rate may affect its concentration, since you may gonna lose the correlation between the concentration in your saliva compared to plasma. So generally you have to study and test, for example, the effect of the flow rate and the pH carefully for each of your compounds, and then move to the sampling approach that it is better in your case. So stimulated or not stimulated, and which kind of sampling device. So whatever the analyte and the matrix, this is the analytical workflow we generally employ. It is made up of three main parts. The first one is the sample pretreatment. Then a microextraction by package sorbent technique is used for the cleanup and analyte concentration. And MAPS is directly coupled to UHPLC MSMS for the targeted profiling of oxylipins and PUFAs. Generally, the pretreatment of the biofluids is required to avoid the occlusion of the MAPS cartridge and to avoid the worsening of analytical performances. So while choosing MAPS extraction, recent trends in sample prep includes automation, includes miniaturization, and low cost and low solvent extractions. And we can say that MAPS shows all these desirable characteristics. MAPS can be considered a miniaturized version of HSPE. It is made up of a syringe and a cartridge that is directly inserted into the syringe. The cartridge is contains the sorbent packet bed that can be of various types. So there are C2, C8, C18, and etc. And it is reusable, so it can be used dozens of times. So MAPS takes lots of advantages. First of all, it reduces to the microliter scale every step of the extraction procedure, and thus ensuring a very high sensitivity in a faster and easier and more environmental friendly way compared to the traditional solid phase extraction and liquid-liquid extraction. And since its development in 2004, around or more than 100 papers have been published reporting the use of microextraction in the field of analytical chemistry and for different kinds of applications concerning, for example, the field of bioanalytics, pharmacology, food science, or environmental science, thus suggesting a very high versatility of this technique. So in our workflow, we employed a C18 cartridge and an automated workstation from HTA that ensure the automation of and the automated extraction of a batch of more or less 20 samples. For each sample, the procedure consists of 5 sequential steps, which are activation, conditioning, sampling, washing, and dilution, which are the steps typical of HSPE extraction. And for each step, the syringe draw up and down or the solvent or the sample for its up and down cycles. So the sample and the solvent pass through the cartridge 2 times. So this is just a warning for all of us when we try to not use these MAPS cleanup after the pretreatment of the sample. So we just pretreat the sample and then we filter, dilute, and inject into the UHPLC MSMS system. just after 10 injections, we observed a very huge increase of the background noise, as you can see in red. And the instrument got dirty. We had to clean everything. So this is just to suggest that trying to choose the best and the optimal sample prep is fundamental to obtain very accurate and very sensitive results, and in particular when working with systems like UHPLC MSMS. So this is the system, the instrumentation we employed. So this is a UHPLC triple quad from Agilent. We employed a C18 column, a Polaris C18 column, and a gradient elution using 0.1% of aqueous formic acid and a 1-to-1 mixture of methanol and acetonitrile. We employed an ESI source in negative mode, and the triple quad acquired the data in MRM mode with 2 transitions per compound. So this is the panel of compounds that we detect simultaneously from each sample. And as you can see, we are covering a broad range of compounds, starting from isoprostanes and prostanoids, up to neuroprostanes and dihomoisoprostanes. This thanks to the collaboration with IBMM and the team of Thierry Durand. We were able to extend our panel of standards to these new families of isoprostanes and neuro and dihomoisoprostanes. And we even detect and quantitate epoxy and hydroxy PUFAs, specialized pro-resolving mediators, and the precursors, all of them in a single run, and all the pure standards were supplied by Kaiman Chemical. So the targeted profiling of all these oxylipins and PUFAs covered a broad range of biological fluids. So they are detected in plasma for sure, but even in dry blood spots, oral fluid, urinary exosomes, and culture media. For a comprehensive characterization of the inflammatory cascade, and to get a very clear picture of what's going on in terms of inflammation, starting from the pro-inflammatory phase, so with all the pro-inflammatory mediators, up to the resolution phase. So these are the details about the procedure for DBS analysis, so dry blood spot detection and quantitation of oxylipins and PUFAs. As you can see here, there's a first step of card pretreatment. So generally we pretreat our filter paper using an antioxidant SBHT to avoid autooxidation ex vivo of the compounds, and we use a mixture of internal standards, labeled internal standard 5 microliters, that were directly spotted onto the filter paper. Then there's the step of blood spotting through a microcapillary device as the mini-vet POC device. And then once dried, the spot is extracted using a mixture of methanol and water, 70-30. The supernatant is filtered and is diluted before MAPS cleanup. MAPS cleanup, as I said, is made up of 5 steps, and in particular, the elution is performed using 30 microliters of methanol that are then directly injected into the UHPLC MSMS analysis without any evaporation step. So when compared to the traditional addition of the internal standard into the extraction solvent, this innovative approach of adding it directly on the filter paper ensures the correction of analyte variations not only during the extraction and analysis, but even during the most critical phases of DBS analysis that are for sure manipulation and storage. So as you can see here, in the case of 15F2-isoprostane, the normalization to the internal standard did not change so much things, because these compounds proved to be pretty stable in DBS, both at room temperature and 4 degrees up to 30 days. But this was not the case of 15E2-isoprostane, for example, for which we observed a dramatic decrease of the concentration just after 3 days of storage. And thanks to the normalization to the internal standard, we ensured an accurate quantitation of the analyte up to 21 days of storage. So these are our recommendations based on the results obtained on the overall panel of our compounds, so on the 60 compounds, not only on the examples I mentioned before. So once you protreat the card, you can use it within 1 month when stored at 4 degrees. Generally, once you spot your blood onto the filter paper, you have to send the card to the lab and analyze it within 7 days by storing it at 4 degrees. And about the methanolic extract, it is stable up to 1 month at -80. So if you want to perform analysis, a double analysis on the same sample, or you have to delay your UHPLC-MS analysis, the extract is pretty stable. So the accurate quantitation is even guaranteed by a volumetric sampling approach, as I mentioned before. So as you can see from the video, the sampling procedure is extremely easy. So we generally discard the first drop of blood to avoid contamination, and then we collect 50 microliters of blood through this microcapillary device, which is loaded with EDTA, and then we spot the blood directly onto the filter paper immediately. And we let the spot dry for 3 hours. So why using a volumetric approach that is less straightforward than the non-volumetric ones? Because we observed hematocrit and drop volume effects for these kinds of analytes. As you can see from the response surfaces, when working and dealing with low volume of samples, so from samples low and spot volumes lower than 30 microliters, we observed a bias of the concentration compared to the reference value that were higher than the accepted bias, that is ±15, as you can see in green, across all the HCT investigated, so from 30 up to 60. When using instead a volume of spot higher than 30, we observe the system works pretty well, but not at the highest HCT level, so from 50 to 60. In addition, we observed a huge chromatographic effect. We observed a lower concentration of the analyte in the center of the spot and a higher concentration in the peripheral area, with biases that were around 30-40% in the case of the analytes and 60-70% in the case of the internal standard. So as you can see here, the internal standard did not mimic very well the behavior of the analyte on the blood spot in the case of this chromatographic effect and this inhomogeneous distribution, and thus we were not able to normalize this effect, and that's why we moved to a volumetric approach. In addition, we test the DBS plasma correlation. This has been tested for the first time in a paper that has been recently published. Here it is, on free radical biology and medicine. This DBS approach has been applied for the monitoring of heart failure patients, but in the paper there are lots of even analytical aspects that have been investigated, as, for example, the evaluation of DBS plasma correlation. In general, we found a good correlation, but not strong, in the case of the precursors, so of polyunsaturated fatty acids, with correlation that were above 0.5. And in particular, this correlation was independent on the level of the hematocrit, so that ranged from 35 to 50, and it was independent on the type of patient or subject we recruited, because this correlation was performed by collecting plasma and DBS samples in control patients, but even in patients with cardiovascular diseases as heart failure disease, thus making this correlation even more robust and reliable. Unfortunately, in the case of oxylipins, we did not generally observe a significant correlation between DBS and plasma sample. In particular, as you can see from the compounds highlighted in yellow, which are thromboxane B2, PG2, or 12-hit, we observed a very high concentration in DBS that are higher than that in plasma, found in plasma, from 10 to 100 in the case of 12-hit. So there is a very dramatic increase in DBS, and it may be related to ex vivo activation of platelets and leukocytes, since all these compounds are COX and 12-LOX derived metabolites. And this is an aspect that needs further investigation. These results suggest that we have to study in detail all the aspects about the ex vivo activation of some of the enzymes that may be present in blood, and that we have to study in detail manipulation and storage to assess eventually the correct correlation between DBS and plasma samples. So it's a very new field. It has never tested before, and it's something that needs further investigation and very strong work. So this approach has been applied for the monitoring of heart failure patients, in particular when they were performing a sacral ergometer test. So that's why we employed a DBS assay instead of plasma collection. Heart failure is a complex syndrome that consists in the inability of the heart to meet the body needs. And it can be treated, but unfortunately not cured. And in this study, we were interested in differentiating the two phenotypes of the pathology, which are HF-ref, or HF with reduced ejection fraction, and HF-pef, or HF with preserved ejection fraction. So we perform a small pilot study on these patients. We collected patients with stage AB, that are patients with no HF and hypertension and diabetes, that are our control group, we can say. HF-ref and HF-pef. Each patient underwent a cardiopulmonary exercise testing coupled to exercise stress echocardiography. And this pilot study has been performed in collaboration with the Heart Failure Physiolab of Santa Chiara Hospital. And for each patient, we collected 3 DBS samples, one before at the maximum of the effort and after 2.5 minutes. So these are the results from this preliminary pilot study that are mainly related to PUFAs. And so compounds for which we observed a good correlation between DBS and plasma samples. As you can see here, at rest, we observed lower level of both arachidonic acid and the other omega-3 PUFAs as EPA, DHA, and DPA, moving from left to right. This means moving from patients that have risk factors as diabetes and hypertension, but not heart failure, up to late-stage HF, which is characterized by this kind of population that is HF-ref. In particular, arachidonic acid showed a significant difference between HF-PEF and REF, so between the two phenotypes of the pathology at rest. And it was particularly interesting the behavior of arachidonic acid during exercise, which decreased significantly only in the case of HF-PEF population. So not in HF-REF, not in patients with risk factors, but only in HF-PEF, suggesting not only a different bioavailability of this arachidonic acid in these patients, but maybe even a different use as energy substrate during exercise. Differences in terms of PUFAs and in particular of DPA have already been observed in a previous work performed on heart failure patients in which we employed saliva as biofluid for sample collection. In that case, this was the procedure. So we collected the saliva samples using salivate, the salivate device. After collection, 0.5 ml of saliva were extracted using a mixture of inorganic salts and acetonitrile. Then the supernatant was filtered and diluted with water before MAPS, UHPLC, and SMS analysis. So the study design in this case was a bit different, not only because we employed saliva instead of DBS, but even because we collect samples coming from acute heart failure patients, so heart failure patients during acute decompensation episodes, so at the admission and at the discharge from the hospital and at the follow-up 1 month later. During the follow-up, we even collected samples from patients with chronic heart failure and with the risk factors as obesity and hypertension. So, as you can see here, we observed in the case of DPA a significant increase of these omega-3 PUFA in the acute phase of HF compared to the chronic phase. Instead, a reduced amount of lipoxin B4, which is a pro-resolving mediators produced from arachidonic acid, a reduced value in at the admission in the acute phase of the compensation compared to the chronic phase, and a sort of increasing trends going back to the levels of the chronic population. Thus suggesting probably that lipids may play a key role in myocardial rearrangement during acute decompensation episodes. So heart failure is one of the age-related diseases, together with other cardiovascular diseases and neurodegenerative disease, and they have in common late-stage chronic inflammatory stages. And how should we delay or avoid the onset of this late-stage chronic inflammation? For sure, for example, performing a regular physical exercise may help us and may help training our body to respond to the onset of inflammation and oxidative stress. We monitor oxidative stress in athletes. In particular, we monitor, for example, oxidative stress in swimmers that were performing a cycloergometer test using saliva samples. Or more recently, we performed the monitoring of athletes, ultramarathon runners performing the Tour de Géant. That is a race of more or less more than 300 kilometers. So it's something so strong. But as you can see here, in the case of these athletes, we observed after the race an increase of 5F2 isoprostanes, but not as, but an increase that was a mild increase, right? When referring to the 300 kilometers they run. This is mainly because these athletes with a regular physical activity train their body to respond to the stressors and so to these stimuli, and they train their antioxidant system and their body to go back to an homeostasis state, as even confirmed by the presence in their urinary exosomes of these specialized pro-resolving mediators as resolving D1, D4, and lipoxin B4. Thus suggesting they are trying to go back to an homeostasis state, and they are able to respond quickly to the oxidant and inflammatory insult. So, unfortunately, we are not all athletes or ultramarathon runners, but we have to take care of this aspect since, unfortunately, we are, during our lifetime, continuously exposed to both endogenous and exogenous stimuli that trigger inflammation and oxidative stress, as, for example, hormones, viruses, air pollutants, or microplastics. When talking about viruses, maybe you will have thought of the recent pandemic. I have to say that I had the chance of getting samples from the first pandemic wave here in Italy. And I have to say that I have never seen such kind of signal related to oxylipins. So we found a concentration of these compounds in the plasma of infected people of around hundreds of ppb or ppm. That's something crazy for compared to the level of these compounds in a normal state. And these oxylipins prove to behave even and to be even more informative than cytokines in differentiating between the mild patients in blue and the most severe in red. And in particular, this difference was based on the levels of specialized pro-resolving mediators that were up to 100 times lower in the most severe patients. In particular, oxylipins proved even to be good in differentiating between the different SARS-CoV-2 waves with the wild type that was characterized by the strongest oxylipin storm. And what about the chronic exposure? Because this was like an acute exposure to an infection coming from a virus, but what about chronic exposure of our body to, for example, micro and nanoplastics that are circulating in our blood? This is a very recent work we have performed on cardiomyocytes. So we incubated cardiomyocytes with microplastics of different types of plastics, and we observed the inflammatory response in these cells after 3 and 7 days. So as you can see here, we observed an increase of the inflammatory response in these cells, as you can see from the overexpression of interleukin-6 and TNF-alpha. We even studied the release of VOCs in the headspace of the cells, and we observed the presence of pentane, propanol, and hexanol, which are key indexes of oxidative stress. And what about the production of oxylipins? So as you can see from these heat maps, we observed an upregulation of oxylipins in the cells incubated with microplastics. So from left to right, so from the control up to virgin microplastics and aged. And that was different from a type of plastic to another. So, and this was the trend of lipoxin before. So it was just to conclude, this was just a brief overview of the detection and quantitation we perform in our research group of oxylipins in unconventional biological fluids. What's next? For sure, standardization and harmonization, as suggested by this important paper, the technical recommendation that has been submitted, thanks to the great job of Niels Scheb and Valerie O'Donnell. And but in the field of oxylipins, and in particular regarding unconventional biofluids, that's more something more to do as definition of the protocols, in particular of manipulation and storage. So I hope I convince you that it is worth it to go ahead and to try to define these protocols and and go ahead with standardization and harmonization. And thank you all.
Okay, thank you very much, Denise. I think the point that you're making about storage and the kind of oxidation artifacts is really important. And Ginger, I'm going to hand the microphone over to Ginger first, because she has a question that relates to this. And then maybe we can discuss this a little bit. So Ginger.
Thanks, Valerie. I was just curious about the dried blood spots analysis and the correlation with the plasma in particular, because the dried blood spots would still be whole blood. So you'd have the presence of compared to the plasma. So do you think that is what you're seeing the difference accounting for there? Have you thought about that or looked at that or thought to compare with whole blood versus the DBS?
Yeah, you broke up there, Ginger, but I think what was in your question was the presence of red cells being the difference, right?
Yeah,
right.
For sure.
Yes.
Yeah, for sure. This may be one of the cause of the difference between what we observed in plasma and DBS. For sure, in plasma, we are not getting the contribution of red blood cells. And the fact is that we would like to use some devices that can fractionate all the different parts. So you can get information about red blood cells and about serum, and we can understand which is the contribution from the different fraction. And in particular, for the compounds I showed, there were such an increase, like 100-fold increase. So in that case, since they were all COX and 12 LOX, it seems like they were all platelet derivative metabolites. So it was something related to ex vivo activation of platelets and leukocytes. But in the other case, it may be something just because we are comparing something different. So there's a contribution of oxylipins and PUFAs, obviously, even in red blood cells.
Yeah, I mean, to follow up on that, I mean, a concern I would have, and it would be good to hear how you've addressed this in your processes, is that red cells can lyse and release hemoglobin, which is highly oxidizing. And as you've pointed out as well, by putting the blood onto this spot, you're getting platelet activation and probably white cell activation also. And if the spot is, stored at 4 degrees for 7 days, there's a huge potential for stuff to happen ex vivo there. So yeah, how do you stop that kind of artifactual autooxidation that hemoglobin can mediate? Because it's really difficult, I think.
It is.
Yeah.
It is. We tried to store at -80. The situation improved a bit, but not so much. So we could extend the stability from 7 to more or less 15, 20 days, but not more. So something happened even at that temperature. And I don't know. Maybe. I don't know, using EDTA that chelates calcium ions may help directly on the paper.
I don't think it's going to stop hemoglobin oxidation. No,
or maybe using some cards that are protected to lyse cells and lyse enzymes, so you can inactivate if it's something that happens because of enzymatic reactions, but.
Yeah. Is it a problem to just take the blood and snap freeze it at -80 instead? I mean, is it the dry blood spot just to make it more convenient? Is that the reason for using it?
Yeah, for example, in the case of heart failure patients, it was the only kind of biofluids we could use. So the blood draw was not the right procedure we could use there because they were performing a cycloergometer test, so it was not feasible. And that's why we moved to the DBS. And in terms of potentials, like for diagnostic purposes, it would be, or for future application to the clinics, it would be the best if we could understand how to stabilize and how to use this kind of
it's tricky.
approach.
I mean, it is. I'm not trying to avoid this, but I think I want to come back to a comment or a question I've got, and maybe Ginger can comment on this too. And that's also something you mentioned about, these other fluids should correlate with plasma. I don't know that would be the case with urine, for example, because what you measure in urine is quite often renal metabolites coming from, kidney metabolism of oxylipins. So I don't know that they would correlate, or why we might expect them to. And maybe it's worth commenting on that, and maybe hearing what Ginger thinks, because she does a lot of urine measurements, actually.
I wouldn't necessarily expect them to correlate either.
No.
Especially for the metabolism aspect and the renal aspect.
Yeah. Have you tried to correlate them, Denise, between urine and plasma?
No, and honestly, I do not measure urine so much. Generally, I've been working with saliva and dried blood spots, but it's not easy to evaluate correlation. So to perform a systematic study of the correlation between these biofluids and plasma, so it's something that we would like to do, for example, for saliva samples.
Okay, thank you. So Tong Shen, you had a question about methodologies. Well, I'll ask the question for you. So the question was, could you elaborate on the MEPS steps technical variation and carryover percentage?
To avoid carryover, we had a final step of cleaning up the syringe. So the entire volume of the syringe is flow with a mixture of organic solvents in order to avoid any carryover effect. So the carryover effect from a sample to another is negligible since we test it before, and we use this kind of approach to avoid it. And as concerns the variability, it is an automated extraction, so the variability is very, very low. If we talk about the entire procedure, we stay within the 15%, but in the case of just of the MAPS extraction, we can say that we can range from 5 maximum to 10%. And even batch to batch, so even the cartridges that comes from a different batch.
Brendan Naker?
Yeah. Thank you, Denise. Great presentation. I just have a comment, to be honest, but you can somehow answer on it. It's actually based on what Ginger asked or said, that there's not enough correlation seen, of course, between the dry blood spot results versus the plasma. But however, when I looked at the table, there were some, interestingly, some correlations. I had very low concentrations, though. So I don't know if you picked that. You've obviously worked with these results and looked at them, scrutinized them. Are there some correlations that we should even think about?
So you mean from the table I showed with the concentration,
Yeah.
the differences
Correct.
between
Yeah, because you
plasma.
highlighted 3 of them. They were obviously very uncorrelated, but from what I noticed, there's quite a few, in fact, at low concentrations that seem to be correlated.
Between the compounds, or you mean the difference between plasma and DBS was lower, because there is a difference between the correlation and the bias that you have with DBS and plasma. So if you have a fixed bias, maybe you will have the correlation, but with a certain bias, right? So the point here is that there's something like artifacts formation, probably ex vivo during the steps of manipulation and storage that change the level from in the DBS, and this makes the results uncorrelated. So for some compounds, this bias between the DBS and plasma is reduced. For most of the compounds is not so big as in the case of COX and 12-LOX metabolites, but it's present.
Yeah, no, I think I was
compounds.
asking more on the ones that correlate, but yeah, go ahead. Val, you have a comment.
Yeah, I think what you're saying, Brendan, is you saw some that were different between the patient groups, right? But I think my worry with that, or what Denise is saying, is that they may have been generated ex vivo. So I suppose then you have to think, okay, they're generated ex vivo, so they really weren't there in the first place. And that probably applies to some of the rare ones she's talking about. But if one sample type generates more of them ex vivo than another, there's some difference there. Like maybe there's a higher level of LOX activity or more hemoglobin around or whatever. We just don't really know what's going on in that ex vivo situation. It's just got to be kind of clearly reported that although they're different and they can stratify the patients, they may be getting generated ex vivo as part of the assay. And I think Niels Scheb had a paper quite recently, which probably may be useful in this context too, where he showed about leaving plasma on the bench. And over the 4 or 5 hours, stuff starts to appear from this category of lipids, from oxylipins that aren't there basally. So ex vivo generation is a big thing. I mean, but nonetheless, it may have its uses if it stratifies patient groups, so long as we know what we're looking at. Yeah. Does that fit with what you're saying, Denise?
Yeah.
Yeah.
Yeah,
okay, good.
Thank you, Denise. Just what is your favorite, saliva or DBS? Because I remember when I just saw the papers on saliva, I say, hey, it's very interesting matrices for patient. And after that, DBS also. But what is the best for you? And of course, oxylipin.
Okay. I started with my journey with DBS, so probably that's why I'm gonna say DBS. But they are both very interesting biofluids, and they have great of potential, but there's a lot to do, a lot of work, and there are lots of aspects that for plasma are already established, as for example the storage manipulation. More or less we have a standardized protocol. In this case, everything needs to be built up step by step. So as for the case of correlation, every idea or hypothesis is welcome, since I'm an analytical chemist. So I really need collaboration and contribution from different types of research area and research field, and so I think that I would go ahead with both of them, working with both. With saliva, we had that stop because of the pandemic. It was not so easy to collect saliva for biosafety reason, but we would go ahead for sure.
It just remains for me to say thank you very much to Denise for her talk today, and everybody for coming, and we'll see you in another month. Okay, so thank you.
Thank you so much.
all.
Bye-bye.